The future of Alzheimer's diagnostics stems from a simple blood test. These blood tests are minimally invasive and have the potential to redefine the way we diagnose and treat individuals with Alzheimer's disease. There is still work to be done to optimize the accuracy of these diagnostic methods, which is why we, in conjunction with Superfluid DX, have been working on developing a process that will determine disease status based on a blood sample. Through data visualization and statistical inference, we can look for patterns in the presentation of genetic data within the blood samples to find apparent trends that could be used to identify and predict the development of Alzheimer's disease within a given individual. This method of diagnostics is more efficient and cost effective than traditional methods. Because this testing method is less invasive and easier on the patient, it is an ideal way to monitor the progression of Alzheimer's disease, as well as further perpetuate the scientific research being done to find biologically significant indicators of both disease status and level of progression within an individual. This semester's work can be broken down into our different subgroups. The classifier team designed multiple machine learning models to understand which gene is related to the presence of Alzheimer's disease. By simply isolating patients' presence of the gene ApoE and analyzing cfRNA or cell-free RNA, our model achieves a successful rate of 86% of predicting Alzheimer's disease. Utilizing various statistical techniques, the uncertainty team identified the optimal type of distribution that represents the trend of genes. Disturbance was introduced to enhance variability and better reflect real-world conditions. Through synthetic data generation, the gene dataset was produced to facilitate more robust model training. Visualizing the data helped the team to see if the model was maintaining an ideal balance and meeting optimization criteria effectively. The biological pathway team analyzed patient data to define the biological functions of genes that showed high influence on Alzheimer's diagnosis. Using common databases for genetic targets of Alzheimer's, differential expression, and principal component analysis, a cross study was performed to see which genes were present in the literature versus patient data and what their influence was on the classifier. There was a go-term analysis performed which took all the genes in our dataset and grouped it into biological functions based on previous studies. The research being done in conjunction with the analysis of these liquid biopsies also holds promise for developing effective treatments that are personalized to an individual's genetic makeup. This is huge because current treatments for Alzheimer's disease are grossly ineffective and only slightly delay the current inevitable progression of cognitive deterioration that is the capstone of Alzheimer's disease. The future of Alzheimer's diagnostics stems from a simple blood test, and our goal is to bring that future to the present. Thank you for your time.